Let me take you inside a cell. Imagine your cell sitting in the body, dormant, it's boring, it's Saturday afternoon, and all of a sudden you're confronted, say you're in the lung, and you're confronted with a virus. You breathe the virus in, and the cell all of a sudden has this task of responding to it and making sure that that you conquer this virus, right? And all of a sudden, the cell is activated. We have cells in our body that are there to do especially that. They get these signals, the cells that on the outer layer, send signals inside. It's really exciting. These cells now get all revved up because, now this is their moment, to come to the forefront. And in order to do all that, what has to happen is we have to have the cell receive these signals on the surface, send the signals through a bunch of proteins, that kind of like doing a race and passing the baton, right? The person at the front end on the cell surface receives the signal, then touches another protein, then touches another protein, and then finally this gets into the nucleus, which is where our DNA sits. And the DNA then has the task of activating the right genes at the right time, in the right place, to now immediately take these genes that were dormant, because we are not always reacting to virus, right? And that would be a huge waste of our energy to be making them all the time. These genes are now activated by these other sets of proteins that are called transcription factors that sit in front of the gene. This is also a little stretch of letters, nucleotides, that are like little signposts that say, if you want to start this gene that's next to me, I need to have the right protein sit on me and tell me what to do. And so these proteins sit on this region, and that's a signal for this gene to be expressed and made into an RNA, which is an exact copy of its DNA, with the simple difference that DNA is deoxyribonucleic acid, and that defines the sugar, and RNA is ribonucleic acid. It's a very subtle difference. But the big thing is the RNA is not stuck in the genome. The DNA genes are all stuck to each other. They can't move. And the RNA has the luxury and the ability to now leave the nucleus, go into the cytoplasm, and then say, hey, I have the instructions on how to kill this virus. So if you want to save this cell, you need to take me, read what I am bringing, the instructions, convert it into an amino acid sequence through translation. And that's really fascinating. It's like blocks, that are chained together, that are reading 3 nucleotides at a time. Our genetic code is defined, it's a 3-letter code. And this is finally made, a chain of amino acid folds upon themselves and is made into a protein that then can, have a function in terms of exterminating this virus. And in fact, when we are dealing with the virus, we make an army of proteins that surround the virus. And, basically, if we are successful, kill the virus. And so the microRNAs are there when we are done with the job. The cell doesn't know enough to stop, because we have now activated a gene that's not supposed to be active when we are not facing a virus. And it's very important we turn it off when we have taken care of the virus for a number of reasons. First of all, we are expending extra energy for no good reason. But more importantly, some of these active genes can turn into cancer and disease forms. So the vast majority of diseases in our body come about through the inappropriate activation of genes in the wrong time at the wrong place. And that's where the microRNAs come. So you can imagine if we had to activate 10 genes to take care of this virus, and after the virus is gone, the cell needs to shut these genes down. We still have the genes making the RNAs. We can take away the transcription factors at the signposts. However, the RNAs are still around. We have to have, in this scheme, where the set of genes that are on and off defines your state at any given time, we not only have to have ways of turning on the right genes in the right time, right place, and turning off, but we also have to have a way of very rapidly clearing their messengers, which is the RNA. And that's where the microRNA comes. So what happens then is the microRNAs are turned on, the right set of microRNAs that will together bind and capture and silence those 10 genes, those 10 RNAs, and take them out to now return the cell back to normalcy. And the binding occurs through, this Watson and Crick base pairing. I told you our genome is a 4-letter alphabet. The letters are A, C, G, T. And the Watson and Crick base pairing says that A's always stick to T's and C's always stick to G's. So, it's really, it's not rocket science. The RNAs know what to do. If there's a specific sequence, that sequence knows which to stick to. And so the microRNAs have sequencers that will bind and capture and stick to hundreds of genes like a zipper. And then once those RNAs are captured, they're degraded and destroyed. So just to summarize, the big role of microRNAs is to allow cells to very rapidly respond to changes. And if you think about it, yes, we need to first establish our bodies, and RNAs are really important. And after that, our survival depends on our ability to quickly respond to, various things that we face. So microRNAs are very, very important.